Okay, so today we're going to talk about Chapter 8 of my book, Molecular Immunity, a chronology of 60 years of discoveries. And Chapter 8 is entitled Regulatory T cells, which I must confess I never even believed in for quite some time when they were first reported by investigators. And so, but this particular chapter is very important in that it brings together a lot of the things we've already discussed in chapters 1 through 7. And it's very important to understand what happens in these regulatory T cell things for what's going to happen in chapter 9, which is immunotherapy of cancer. Chapter 8 actually goes back to what I feel is sort of the beginning of modern immunology in the 1960s. And in 1962, 61, 62, and then throughout the 60s, there's a fellow by the name of Jacques Miller, who was an Australian working in London. And at the time, if you can recall, Sir MacFarlane Burnet had just published his theory, the clonal selection theory, in 1957, and then 59 he published a monograph about this. And so everybody was talking about the fact that the immunology really consisted in 1960 as antibodies. That was it. And everybody really loved antibodies. But there were these other cells that ultimately became T cells that people saw, under the microscope and taking them out of different organs and so forth. And one of the organs that they took out of the experimental animals was the thymus, which is the organ that's in the upper chest. And the thymus is a collection of lymphocytes that lie over the great vessels, the aorta, as it exits the heart and goes to the rest of the body. People took out the thymus, wondering what these cells were. When they looked for antibodies in the thymus, they didn't really find very many antibodies in the thymus, but it was clear that those cells in the thymus looked exactly like lymphocytes. So they figured that it must have something to do with immunity. So they took the thymus out of adult animals and nothing happened. Animals seemed to be just fine, and their immune systems seemed to be working okay as far as they could ascertain at that time. But Jacques Miller, he wouldn't accept the fact that the thymus was just there, was whatever. And so he thought, well, perhaps what the problem is with the thymus is that it's important for the early genesis of the immune system. And so what I should probably do, he said to himself, is that I should take the thymus out of newborn mice. Well, you've probably never seen a newborn mouse, but a newborn mouse is only this big. It's just the end of your little finger. And not even, that's like two newborn mice. And a regular mouse is only about this, 1.5 inches long. So to try to take the thymus out of that newborn mouse, it was quite a feat surgically, and keep it alive. I could probably take it out and kill it at the same time. But he devised a way to go about it, and he perfected his technique, and he started thymectomizing newborn mice that he said that he tried to do it within 3 days of birth, so that the result of his experiments was that when he thymectomized the mice, the newborn mice did fine. They looked beautiful, they looked normal, they grew normally for the first 4 weeks. But thereafter, in the second 4 weeks of life, and as you recall, the lifespan of a mouse is only around 2 years, and a mouse undergoes puberty when they're about 5-6 weeks old. So just about before puberty, the mice started to not look so good. They started to have ruffly fur, they stopped growing, they started losing weight, and in the whole second month of life, they ultimately sort of went down the tubes and died. And so that was interesting. And so because of the fact that Miller was interested in the immune system, he did experiments particularly on the animals in the first 4 weeks of life. And he found that they seemed to have the normal numbers of lymphocytes that compared to he sham thymectomized control mice, newborn mice, and they seem to be pretty equivalent there. But then over the course of the 4 weeks, they gradually lost lymphocytes. They became, and by 4 weeks of life, they became lymphopenic. And when he looked for antibodies, he immunized the mice with different agents, and he found that they didn't make very much antibody or make it very well. And then he thought, well, let's put skin grafts on these animals. And he put skin grafts on, and then the grafts took. The mice didn't reject the skin grafts, even if he put xenografts on, meaning that they came from another species, like a rat. They took too. And so there was clearly an immunological defect in these animals. And so that's what he was looking for, and he reported that. But he also reported in the second 4 weeks of life, when these mice started to become runts, and they looked like they had graft versus host disease. This is exactly what happens if you take a mouse, an adult mouse, and then you give it a graft, a bone marrow graft, or immunocompetent cells from another strain. You'll get graft versus host, and you'll get host versus graft, and you have a big battle going on, and the mice start to become runted. They're not healthy, and they just have an ongoing immune reaction the whole time. So he looked at these mice, and lo and behold, the mice that were lymphopenic in the first 4 weeks of life, now they started to have activated lymphocytes all over their bodies, infiltrating all of the organs, and autoimmune phenomenon and autoimmune hemolytic anemia. They started making antibodies to their red blood cells. They clearly were not doing well. And so this was really a paradox. Because how can you be immunodeficient and then have hyperimmunity or autoimmunity at the same time, or sequentially in the same animals? Well, that was where it sat for 20 years. And if you recall back in between 1960 and 1980 that was the 2 decades of cellular immunology, when people were taking the cells out of the animals, growing them in their cultures, and doing all kinds of mixing and matching and doing experiments. And lo and behold, along came a Japanese scientist, Shimon Sakaguchi, in 1982. He published a paper in the Journal of Immunology, and he said that if he took day 3 thymectomized mice, or nude mice, and nude mice were found to have a genetic defect so that they didn't make a thymus. They didn't have a thymus, and they didn't mature T cells. Either the day 3 thymectomized or the nude mice. If he reconstituted those animals with T cells from adult mice, hoping to make them better again, he found that if he took the so-called helper T cells out of the adult T cells that he transplanted into these mice, that he got a phenomenon that was very, very similar to what Jacques Miller had reported 20 years previously. He then hypothesized that if you did the thymectomy and early on, just like what Miller had found 20 years earlier, that there was a critical T cell that didn't mature and didn't go into the periphery and didn't populate the animal, so that it was normal, so that the immune system was normal. And he hypothesized that this lack from the helper T cell compartment that he had transplanted, when he didn't have those cells there, that perhaps there was some sort of a suppressor cell or a regulatory cell that was necessary to prevent this hyperproliferation and autoimmunity and all these other different kinds of things that were happening. And he coined the term regulatory. There must be a regulatory T cell. At that point in time, that's where it sat in 1982, and thereafter he repeated his experiments and extended them in 1985, and then we go all the way to 1991, 5-6 years later, and then Ivan Horak, who was an investigator in Germany. It was just at the time when people could for the first time knock genes out of mice and have them grow up without you could take the gene out. And then the question is, what would happen? Ivan decided to knock out the interleukin-2 gene. Surprise, surprise. And what happened was, when he did that, just like Jacques Miller's animals, for the first several weeks, the mice were born. They did well. They gained weight. They grew. And if they did experiments in his first paper, they took cells out of took spleen cells out and so forth, and he activated the cells in culture and to ask the question: do they seem to respond normally? Can they proliferate and do all these things? And the answer was, well, there was a marked diminution, 70% diminution of their ability to respond to different stimuli and so forth when they couldn't make IL-2. And I sort of thought, well, okay, but what they've wondered and they thought was, well, why did you get any at all? Because Kendall Smith and all of his cadre were going around the world saying that this was the universal T cell proliferative agent and so forth. And so they, without the interleukin-2 gene, shouldn't have any proliferation. Then what happened was it took a couple of years, and down in Zurich in Switzerland, there were a couple of investigators that were very proficient in studying infectious diseases in mice. And their names were Hans Hengartner and Rolf Zinkernagel. And so in 1993, Ivan Horak got together with them. Actually, before that, I should probably say that I went over, as soon as I found out about this, I got on the next plane and I went over to Germany, and I said, so tell me about these experiments. And they led to some collaboration, and I'll tell you about it in a few minutes. And so I knew all these people. I knew Hengartner, I knew Zinkernagel, and so forth. And of course, Zinkernagel and Hengartner they were in the camp that didn't like these cytokines. This is lymphodrek. And so they didn't want to have to deal with them as being important for the immune response. Horak got together with Hengartner and Zinkernagel, and they did experiments. They took these IL-2 knockout mice and they infected them with the different viruses and so forth. And they found that their immune response to the different viruses was diminished, but the animals didn't die. And so they published a paper in Science, no less, one of the most prestigious journals. And their interpretation of their experiments was that it was obvious that interleukin-2 was not really, there are other it was one cytokine, but there was a whole bunch of other cytokines could just take its place, whatever. Then following that, a friend of mine, Christine Byron at Brown University, did better experiments and more extensive experiments and with more animals. The one thing I didn't like about the Hengartner and Zinkernagel paper was they published these graphs and data things, and they're one animal per point. That's just never done. You usually have at least 3 and sometimes 5 or 10. So you get an average, say your experimental and then your average of your control, and you see whether they're different. Zinkernagel, they didn't have very many mice because they were growing these very precious IL-2 knockout mice, and so they just had one animal per group or per point. Well, Christine did a much better set of experiments, and she infected the mice with exactly the same viruses that Zinkernagel and Hengartner did. And she found that the generation of cytotoxic T cells was diminished by 90%. I was much happier with that result, but I would have been even happier if it had been gone altogether. So that 10%, any 10 to 30% reaction is important as time goes on, and we'll find out about that. Well, then we got together, Horak and I got together, and we decided to see whether or not if we treated these mice with interleukin-2 from the time that they were born and then on, whether or not you could prevent this runting disease and immunodeficiency and lymphoproliferation. That's what they had, in exactly the same phenomenon that the thymectomized mice did. And what happened was the answer was yes. As long as you continued the treatment of these mice, and we had to give them daily shots. And you can imagine these poor little mice, only as big as your little fingertip, and sticking a needle in their tummy and squirting a tiny little bit of IL-2 into them. Anyway, they grew up, they were fine, they did well. And then if you stopped the interleukin-2, about, I think it was like 10 days later, they started to go downhill and die. And so it was clearly you needed IL-2. And so IL-2 was a very important molecule. And the question is, well, why is that, and what does it do, and so forth. The next thing that happened was, around in that same time period, was a mouse had been floating around in the immunological circles for quite a while. This mouse was called a Scurfy mouse. And it turns out there was a mutation in a mouse colony in Oak Ridge, Tennessee, where the national laboratories were when they established them during and after the war as for the atomic energy business and so forth. So they had these mice there. There was a mouse noticed that it was sort of funny looking, and they were able to propagate that mouse. And 10 years later, in 1959, they did a series of genetic experiments, and they found out that wherever the mutation was in these animals, it was in the X chromosome. That's the males have an X and a Y, and the females have two X's. So it turns out then that only the males were afflicted by this, and it had exactly the same kind of syndrome as the day 3 thymectomized mouse. It looked okay for a while, but then as time went on, they started to get scurfy. So that's probably why they were named Scurfy. And so it was decided that they had an X-linked, or it was shown that they had an X-linked recessive mutation. And these mice would go downhill very rapidly and they got really sick and died within 3 weeks of birth. Very dramatic. And when they looked in these mice, they had lymphocytes infiltrating all over the mouse, just like in the other ones that I've just discussed. Same kind of syndrome. They got autoimmune hemolytic anemia, antibodies to their red blood cells. And I should point out that this is exactly the same kind of a syndrome that the CTLA-4 knockouts that I talked about in the last video, they got the same kind of a syndrome. So there's something funny going on here that's trying to tell us something is the thing. Then in 1991, it took about 30 years for immunology to catch up to these mutations, or they had this mouse figured out in 1959, 1960. So it wasn't until 1991 that we knew enough about cells and molecules and T cell receptors and all this other stuff. Interleukin-2. And there was a woman by the name of Virginia Godfrey who got together with the people who had originally described the genetic defect, the geneticists at Oak Ridge. And she found that the primary defect was in the CD4 positive T cell compartment. And of course, that's important because this is the cell that makes the most interleukin-2 on the one hand, and it's also the cell that Shimon Sakaguchi had already said, there's something in this compartment, in the CD4 compartment, that is problematic. Well, then they did a whole series of experiments, and Virginia Godfrey did in 1995 and 1996, and said that these if you took the cells out of the mice and put them in vitro, they made huge quantities of cytokines: interleukin-2, interleukin-4, interleukin-5, interleukin-6, interleukin-7, 10. Gamma interferon, which is the macrophage activating factor, TNF. These mice are loaded up with pro-inflammatory cytokines as well as the so-called immunoregulatory cytokines. The macrophages were putting out huge amounts of colony stimulating factor for macrophages, and they were expressing large quantities of the B7 molecule on their surface that activated the co-stimulator in T cells, the CD.8 molecule. If you put the cells in vitro and didn't give them anything to stimulate them, they proliferated just fine without that. They didn't need to be stimulated. And so at the end of that series of experiments, they postulated that the problem in these mice and the mutation was that there was some loss of a normal down-regulatory factor that was involved in T cell activation. And so the question is, what was that? Seemed like a reasonable, that's the reasonable working hypothesis. So the other thing is that all four of the mice that I've talked about, the CTLA4 knockout, the day 3 thymectomized mice, the IL-2 knockout, and now these Scurfy mice, the bottom line is that it looked like there was a common molecular mechanism that accounted for this weird paradoxical immunodeficiency in the face of hyperimmunity. Then, almost at the same time, Sakaguchi came back into the fray again in 1995. And originally back in 82, he had thought that it was a helper T cell that was a regulatory T cell. And he did a series of more experiments in 1995, looking at the same phenomenon, using his mouse model of a day 3 thymectomized mouse or a nude mouse and so forth. And he was reconstituting these mice with different subsets of T cells from normal mice. And he found that if he excluded normal CD4 T cells that also expressed the interleukin-2 alpha chain receptor, that then those mice got into deep doo-doo. They had a major problem. So that he had sort of narrowed it down, because in a normal mouse, mostly non-activated, non-antigen activated T cells, both CD4s and CD8s, when we first described the IL-2 receptor assay, and then the alpha chain of the IL-2 receptor, and then the beta and the gamma and so forth, it looked as though that, and I went back and looked at recently, or when this stuff came out, and initially we found that there were very few of, if at all, any cells that had, without any activation of the T cell receptor, that expressed the IL-2 receptor. They were IL-2 receptor negative. But if you went back and looked at normal mouse cells, about 5% of the CD4 positive T cells had the IL-2 receptor alpha chain. By comparison, if you looked at the CD8 positive compartment, less than 1%. There were some positives, but it was very, very few. So I assumed, I said, well, those are, they're probably antigen activated cells in the mouse, because the mouse growing up in a normal environment is going to have activated T cells in it. However, once he got onto that and it reported that, the field went bonkers, because particularly these immunologists, I didn't tell you about the fact that back in the 70s, there was a phenomenon that came and went that was called a suppressor T cell. And everybody was excited about suppressor T cells, because if you wanted to make the immune response better, you took the suppressor T cells out. Or if you wanted to dampen down the immune response, you gave them the suppressor T cells. And all kinds of experiments were reported, and everybody got it, could do something. Well, it turns out that when they finally identified the T cell antigen receptor and so forth, and then the MHC molecules that I talked about in one of the other videos, they couldn't find the markers that were purported to be markers for the suppressor T cells. There were none there. And so all of a sudden the whole field walked away from suppressor T cells, and nobody talked about them. They were embarrassed that they published some of their best papers in the best journals. So now, fast forward 20 years later into the 90s, some of the young students had never even heard of suppressor T cells, but the old guard said, yeah, whatever, we've been through this before, right? And I have to say, I was one of them. And so, Sakaguchi had a corner on the market in 95 and going forward, because he had the in vivo assay. But the problem with the in vivo assay was it was very cumbersome, and it took a long time to get results, and he was really the only one that could do anything. So along came a fellow from the NIH, Ethan Sheback, who devised an in vitro assay for the so-called regulatory T cells. And briefly, basically what he showed is that if you took just normal T cells and activated them in culture through the T cell receptor with anti-CD3, in the presence of antigen-presenting cells, macrophages, that was important, And then if you threw in your regulatory T cells, but they had to be there in a pretty high concentration. Anywhere from 10% up to 50% of the total cells in the culture had to be these so-called regulatory cells. But if you did that, and then if you measured the proliferative capacity of the normal T cells, you could get a dose-dependent regulatory T cell dose-dependent diminution of the proliferative capacity of the effector cells. And on the basis of his assay that he published in 1998, he made several claims in this paper that turned out to be true, but that raised a lot of eyebrows. One of them was that the regulatory T cells themselves could not make interleukin-2. They were so-called anergic. That became important. The second thing was that he needed the antigen-presenting cells in the cultures in order to show the suppression. And the third thing was that he needed a fair number of these, a fair proportion of these guys in there. That was 98. So we're coming up into some more modern times. And in 2001, there was a splash of papers, some from the Scurfy Mouse people, the original people, and also from people who had been dealing with congenital immunodeficiencies. And they found, the Scurfy mouse people found a mutation in a gene that was called FOXP3, FOXP3, which codes for a transcriptional regulator. So it's a molecule that binds to DNA, and it can either turn up genes or can turn them down. It can do both ways. And there's a whole family of these, and so this is number 3, but there's 1, 2, and so forth. And the Scurfy mice had a mutation in that gene, and that stimulated the people who were looking at poor little kids that had very severe immunodeficiency in the face of autoimmunity, this paradoxical kind of a syndrome. And they looked at that same gene, and lo and behold, it was defective in a set of these patients. Well, that sort of was a shot across the bow, because immediately the regulatory T cell people went and figured out that basically this was the gene that would identify regulatory T cells. And before that, for at least 5 years, since Sakaguchi had shown that they were IL-2 receptor alpha chain positive, that was the only way you could identify them. But the problem with that is that also normal activated T cells would be IL-2 receptor alpha chain positive, and it really wasn't a very good marker to be able to identify them and separate them out and so forth. So now that just added another gene to the mix of CTLA4 and IL-2, and now we have FOXP3, and it looks like we're getting, this whole thing is coming together. And so you have to remember now, this started in 1962 and then to 1982, and now we're up into 2001 and 2. And so that's 40 years it's taken to sort of figure this whole business out. And it gets more, it gets more Byzantine. And I guess that's one of the things that you learn about biology, in particular in immunology. I mean, it's really complicated. That's why I always say to the physicists, I said, what are you guys? This isn't complicated at all. You got one sun, you got one moon, a couple of 3 planets running around. That's easy. Anyway, so in the end, as a consequence, around 2001 and 2 and so forth, and then up until 2003, I started to look at this carefully. And I'm saying to myself, Kendall, you shouldn't be so skeptical all the time, because it looks like it looks like what's going on is they showed early on that FOXP3 was one of the regulatory genes that turned down NFAT, which was, if you recall, NFAT was one of the transcription factors that turns on the IL-2 gene. Well, if FOXP3 turns it down, if you don't have any FOXP3, you're not going to turn down that NFAT, and you're not going to turn down IL-2 kind of thing. So it looked like, and this is when I thought to myself, and this is when I recalled essentially my mentor back in the early days and when I was at Dartmouth, who told me that I couldn't really think about IL-2 as a hormone because there was no negative feedback regulation. And at the time, I tried to do experiments to see if I could find that, and I couldn't, and so forth. So I couldn't really beat the drums as much as I had wanted to back in the early days, that this is really a hormonal system. Well, now, what are we talking about? We're talking about 25, 30 years later, we finally get into that whole process. But now, the regulatory T cell people didn't want to have to say, well, it's all has to do with IL-2. Because that IL-2 was old hat by then. They didn't want to have to. They had their new phenomenon, and they didn't want to give it up for anybody, particularly Kendall, I have to say. So then the question became at that point in time, so what is the molecular mechanism so we have the Treg cells. We think we know in some respects how they work, but we don't really understand how they suppress, how they're the new suppressor cell. And one obvious thing, and that had to do back to the in vitro assay, when you have to throw in 50% of the cells into the mixture have to be these regulatory T cells. And we knew that they were IL-2 receptor positive because that's how Sakaguchi defined it. And it turns out if you quantify the IL-2 receptor alpha chain on those cells, it's like 10x or 100x that you see on the effector cells. And so in 2007, Pushpa-Pandey, and so the obvious hypothesis is that, well, it's these regulatory T cells are just sopping up all the IL-2, and they can't make any of their own. And so they're just binding it and internalizing it and degrading it, and they're taking it away from the effector cells. And so there was another group in the same department at the NIH as Ethan Chivak, who had devised the in vitro assay. And this other group was led by Mike Leonardo, and he had a postdoc by the name of Pushpa-Pandey, and I know both of these people, and they're very good scientists. And Pushpa said she was just young, and she said, well, it looks like it's just an IL-2 sink, and just sopping it up. So she did a series of experiments that were absolutely definitive from my view, that said, yes, that is happening. And if the regulatory T cells sop up enough of the IL-2, then the effector cells actually die. It's not that they just stop proliferating, they actually die. And that was hard for the regulatory T cells to tell people. They didn't want to have to say, well, that's what it is. It's just, regulates IL-2, and because then the story would be over. They couldn't publish any more papers or write any more grants and stay in the game, essentially. And then I got together with a group at Sloan Kettering about that time. And I actually had Pushpa-Pandey come to New York. I was at New York by then, and I had her come and give us a seminar, and I talked to her. And after I did that, that was when I decided that she's a very good scientist, essentially. So then there was a new fellow that came to Memorial Sloan Kettering across the street from Cornell at the time, and his name was Grégoire Altan-Bonnet, and he was a Frenchman who had been a physics major in college in France, and had, after his undergraduate training, had gone to the Rockefeller and gotten a PhD in biophysics. And then he had gone to the NIH and did a postdoc in a cellular immunology lab in the same department with Ethan Chivak and Pushpa-Pandey and Mike Leonard and Mike Leonardo. And so, this is a little bit of an incestuous family that is all together there. And he felt, as I did, and as Pushpa and Mike Leonardo felt, that it's just sopping up IL-2. And but this time he was a very accomplished cellular immunologist, but he was also very unusual because he thought like a physicist and a biophysicist. And so he was interested in not only in molecules, but in forces, the way physicists would do. And so he had a postdoc from Israel by the name of Oner Feinerman. And Oner and Grégoire started doing some experiments, and they talked to me about them, and I started talking to them about them, and I helped them as much as I could, but they really did all the experiments. sort of a tag along at that stage. But they showed that the density of IL-2 receptor alpha chain on activated regulatory T cells, or ones that already had the receptor to begin with, would just go up huge, 20-fold X. And it wasn't surprising to me because we'd already seen that way back on we didn't know about regulatory T cells, but back in the 70s, we first saw that. They showed with a series of experiments that how the quantification of these molecules, the receptor molecules, the IL-2 molecules, and so forth, really explained why and how powerful these cells were as regulators, because of the fact that they could just sop up this stuff as fast as it could come out from the effector cells. So they started talking about IL-2 scavengers. So regulatory cells. That's a pretty. It's not a bad word, a regulator, except unless you're a Republican. But a scavenger, a scavenger is sort of a bottom feeder kind of a thing, and I always liked that term for the regulatory cells. Then another person came into the mix by the name of Kaya Wing, who was a young Swedish girl. She went to the university, graduated, and did a postdoc, and then also decided to do a postdoc with Sakaguchi in Japan. If you know anything about the gender discrimination in Japan from male and female, this I said to myself, I'd never heard of that. I never heard of Westerners going to do it. I heard of some, but not very many, and particularly not women. But she went there and she did really, really good work. And one of the other things that characterized the regulatory T cells, and Sakaguchi had shown this, was that they also had a high density of IL-2 receptor alpha chain on their surface, but they also had a very high density of CTLA-4 on their surface. And if you remember back when we were talking about CTLA-4, the normal cells, the so-called effector cells, they would not express CTLA-4 until they were stimulated through the T cell receptor, the CD28 molecule, and ultimately IL-2. Because remember, Marie Louisa Allegra showed that IL-2 knockout mice couldn't express CTLA-4. So here we have these regulatory T cells that can't make IL-2 themselves, that have a high amount of IL-2 receptor and a high amount of CTLA-4 on their cell surface. So it's coming together now, when we're in the 2000s, and it wasn't until she was with Sakaguchi in the 2000s, the aught years. She then went back to Sweden and went and got a job at the Karolinska Institute and set up her own lab. And then I visited her. I can't remember when it was, but it was within the last several years. And I met her and I talked to her. And again, I said to myself, this one's a good one. It's sort of like when you're going fishing and you pick up a fish. Oh, this is a good one, So anyway, what she did a whole bunch of experiments and published some nice papers about regulatory T cells. But one of the most important ones, I think, was she made a so-called conditional knockout of the CTLA-4 gene. And that is, the way you do this is that you construct a gene that has a regulatory region upstream from the gene, that if you activate that with a drug, it'll splice out the gene. So you can let the mouse grow up and be normal. Remember, the CTLA-4 knockout would get sick and die within 3 or 4 weeks. So you really couldn't do much for that. So she could let the animals grow up and then give them a drug, and the CTLA-4 gene would be knocked out. And when she did that, she found that after you did that, a few weeks later, you got exactly the same syndrome that you would have anticipated when you didn't have any CTLA-4. That becomes really important because of the fact that CTLA-4 then is is a part of the whole interleukin-2 induced negative regulatory feedback loop. And as in my last video, I said, without IL-2, because it's the conductor, you don't get any co-stimulation and you don't get any co-inhibition. You don't get anything. And so you got all these crazy things going on at the same time. And so that's really the end of the story. But in addition to that, I want to read you the last paragraph from chapter 8, because to me this is an extremely important phenomenon that the whole thing is come into being. And I think that the systemic autoimmunity, so that you have what the kind of a syndrome that these animals got when they didn't have one of these genes, is that you lose the negative feedback cytokine control over this whole process. So it's a very sensitive, intricate, positive and negative thing going on in terms of generating an immune response. And so I say in the last paragraph, I say, thus, after more than 3 decades of elucidation of the molecular mechanisms regulating immune responsiveness, it is now evident that regulation of the exquisite determinative capacity of the immune system to recognize and react to non-self, but do not react to self, lies predominantly at the level of negative feedback control of the reaction mediated by IL-2 and other cytokines, rather than the antigen recognition aspect of the system. And so it's to be especially noted that this represents a sea change in thinking about how the immune reaction is regulated, and is contrary to Burnett's clonal selection theory, which stressed the deletion of self-recognition lymphocyte clones as responsible for the lack of self-reactivity. Of course, back in those times, cytokines were still yet to be described and discovered during Burnett's time. And also this change in understanding of how the immune system is regulated has obvious therapeutic implications, because if you know how this autoimmunity is happening, then you know what to do. And so I'll stop there.